Assalamu alaikum. My name is Isha Shaukat and I am a student of DVM. Welcome to this video presentation where I'll be explaining RNA, its types and about its structure. So what basically RNA is? Ribonucleic acid is a kind of nucleic acid found in all living organisms, and its primary function is to act as a messenger transmitting DNA significant instructions. Unlike double-stranded helical structure of DNA, the RNAs are single-stranded. The four major bases in RNA are adenine, guanine, cytosine, and uracil. RNA is composed of a linear polymer of ribonucleotides joined by 3,5 phosphodiester bonds. Three primary, or we can say major types of RNA molecules are messenger RNA, transfer RNA, and ribosomal RNA. And all of the below are involved in the process of biosynthesis, although each differs from the others by size and function. First, I'll explain messenger RNA. Messenger RNA, mRNA, plays an important role in the transcription of DNA. Transcription is the process in protein synthesis that involves copying the genetic information contained within DNA into an RNA message. The mRNA comprises only about 5–10% of total cellular RNA. mRNA is synthesized in the nucleus as heterogeneous RNA, which are processed into functional mRNA. So, it is first synthesized in the nucleus as heterogeneous RNA. The mRNA carries the genetic information in the form of codons. So, this is the structure of mRNA. In the 5' end, there is a presence of one capping with the help of 7-methylguanosine triphosphate. So, this capping is going to help in the recognition of mRNA in the translation process as well as in the capping. It also protects the mRNA from the attack of various exonuclease enzymes, and over here there is an unusual linkage present that is called 5' phosphate linkage. With the help of these 5' phosphate linkage, this 7-methyl can attach to the messenger RNA, while at the 3' end there is a presence of multiple adenylate residue, and this adenylate residue gives rise to the poly tail, and it helps in stabilizing the mRNA structure as well as protects the mRNA from the attack of various exonuclease enzymes. Now transfer in tRNA. The transfer RNA, tRNA, plays an important role in the translation portion of protein synthesis. Its job is to translate the message within the nucleotide sequences of mRNA into specific amino sequences. Transfer RNA is shaped like a clover leaf with 3 hairpin loops. So, tRNA molecules vary in length from 74 to 95 nucleotides. In eukaryotic cells, 10–20% of the nucleotides of tRNA may be modified and known as unusual nucleotides. For example, first dihydrogen D, in which one of the double bonds of the base is reduced. Second, ribothymidine, in which methyl group is added to uracil and to form thymine. Third, pseudoridine, in which uracil is attached to ribose by a carbon bond rather than a nitrogen bond. This is the image showing the structure of ribothymidine. Over here you can see methyl group is added to uracil. Next, dihydroridine, double bond uracil are reduced, and in pseudoridine, uracil is attached to ribose with the help of carbon-carbon rather than nitrogen bond. Now structure of tRNA. So this type of RNA is generally a single-stranded transfer RNA molecules get folded into a structure that appears like a clover leaf. All tRNAs contain 4 main arms. First one is the receptor arm, second one is the D arm, third one is the anticodon arm, fourth one is the PSU arm. The tRNA molecule has a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leaved clover. One of these hairpin loops contains a sequence called the anticodon, which can recognize and decode an mRNA codon. Each tRNA has its corresponding amino acid attached to its end. When a tRNA recognizes and binds to its corresponding codon in the ribosome, the tRNA transfers the appropriate amino acid to the end of the growing amino acid chain. Then the tRNAs and ribosome continue to decode the mRNA molecule until the entire sequence is translated into a protein. Here you can see the 3-dimensional structure of transfer RNA. Function of tRNA. The main function of transfer RNA is that it carries amino acid in an activated form to the ribosome for the protein synthesis. Ribosomal RNA is a component of cell organelles called ribosomes. A ribosome consists of ribosomal proteins and ribosomal RNA. Ribosomes are typically composed of 2 subunits, a large subunit and a small subunit. Ribosomal subunits are synthesized in the nucleus by the nucleolus. Most abundant RNA is around 80%. A ribosome is generally a cytoplasmic nucleoprotein that acts as a machinery for the synthesis of proteins. The ribosome is a spheroidal particle and is composed of a large and a small subunit. So this is the structure of ribosomes. Eukaryotic cell and prokaryotic cell both are composed of 2 subunits, where one is the large and another one is the small one. In the eukaryotic cell, the large subunit is around 60 Swedberg unit. This unit is a non-SI metric unit for sedimentation coefficients, and the small subunit is around 40 Swedberg unit. Each subunit has multiple proteins and RNA particle. So the large subunit contains 28 Swedberg rRNA, 5.8 Swedberg rRNA and 5 Swedberg rRNA, while in the 40 Swedberg unit there are 18 Swedberg rRNA. Now in the prokaryotic cell, the large subunit is around 50 Swedberg unit and the small subunit is around 80 Swedberg unit. Each subunit also has multiple proteins and RNA particles as mentioned in the slide. These are the functions of ribosomal RNA. It is necessary to maintain ribosomal structure and also participate in protein synthesis by binding of mRNA to ribosome. Recent studies suggest that ribosomal RNAs may also provide some of the catalytic activities and thus is an enzyme, ribozyme, which helps in the translation process. These are the other nuclear cytoplasmic RNAs. Besides mRNA, tRNA and rRNA, eukaryotes have some other RNAs. These are first heterogeneous RNAs, second small cytoplasmic RNAs, and the third one is small nuclear RNAs.